Hello, greetings from Malaysia. I'm Rosani from UST Putra Malaysia, and today I'm going to share with you on my journey from Europe to back home, especially on exploring the in silico toxicology from a Malaysian perspective. I have four main things to talk today. The first one is regarding my research journey. Then I will explain a bit more on the in vitro in silico approaches in risk assessment and challenges as a junior scientist and as a toxicologist in Malaysia. And the final one that I'm going to share is about my current work and a way forward. So this is the summary of my research journey. So if you can see from the slide, actually I studied in the Netherlands in 2013, but the research journey itself is not only during my PhD years, but it's actually way before my PhD years, where I started with my project during my high school. During my high school, I participate in a competition of science and innovation. So we call this project as a Hempadu Herbs project. So what we did was we used this Hempadu bumi. This plant has been used traditionally to reduce the blood glucose level, especially with the patient with diabetic mellitus. And what we did was we sample the plant, and then we transformed this plant into capsule, and we gave this capsule to our participants, or our participants, and then the participant, we measure the blood glucose level before and after they took the capsule itself. And then with this project, we were awarded as a young scientist award. So I think this is a starting point when I started to learn about research, how to conduct research, how to to think critically, even though this is really, if I think back, this is really not ethical at all. This is when I fell in love with toxicology, where during my bachelor degree, where I did a research for my final year project. I work with this plant, which is known as ketum or Mitragyna speciosa. So this plant has been banned in Malaysia due to the psychotic effects. So we got the sample from Institute of Medical Research, and I evaluate the mutagenic and anti-mutagenic effects using AMS test. This is the outcome of my final year project during bachelor years, where we were able to publish the research in the scientific journal in 2011, and this is my first research article published. After the first degree, I continue with my master project, which entitled the risk assessment of local shrimp paste intake. So during this time, I sampled the shrimp paste from different places in Malaysia, and I run the mutagenicity assay, and at the same time I also analyze the heavy metal content in the shrimp paste. Then at the same time, I also conduct an epidemiology study to know the daily intake of shrimp paste among Malaysian population. With all this data, then I run the risk assessment. And I was lucky during this time. I got a travel award to attend an international conference of environmental mutation in human population in Qatar in 2012, where I presented my work. And I was chosen to be the first runner-up for the best poster presenter during this conference. And I think this is really boosts my motivation to do more in toxicology. Then I continue my study in the Europe, specifically in the Netherlands, where I work in integrated strategy of the assessment of kidney toxicity, the case of aristolochic acids, where this project has been supported by Malaysian government, and at the same time I was also a tutor at University Putra Malaysia. I was a tutor since my master's degree. Plant food supplements or herbal medicines has been used widely in the population, and one of example they use these plant food supplements is to reduce weight. So this is what happened in Belgium in around 1990s, where a lot of young women took this supplement supposedly to contain the Stephania tetranda, but it was accidentally exchanged with aristolochic because both of these plants use the same Chinese nomenclature, which is Fang Qi. In less than 10 years, many young women suffered from the aristolochic acid-induced kidney tumors. And based on this graph, what you can see from this, these are the cases of Chinese herbal nephropathy or aristolochic acid nephropathy worldwide, where we can see that we have many cases in Belgium, and not only in Europe, we also have cases in the US and China, and a lot of question marks here. And this is actually a worldwide problem. So as a toxicologist, so we need to understand how does this chemical cause toxicity. So upon the exposure to this aristolochic acid, so it will undergo the metabolic activation and it will change to the adduct, DNA adduct, and this DNA adduct are really persistent in the kidney, and that lead to the mutation and cancer. So this is a metabolic pathway of aristolochic acids, where we need to understand how much from this parent compound will convert to the adult, and also it will, it will excreted from the body. So the first task during my PhD project was to use this in vitro model to predict what happened in vivo. So my focus was on the kidney toxicity, and then how to use this in vitro data to predict the in vivo situation. So we used the physiologically based kinetics model based reverse dosimetry, where we estimate how much the aristolochic acids that goes into the system, and then we model it in different model. For example, we have model in rats, we have also model in mice, and also we also have model in human. So based on this data, where we measure the point of departure for a risk assessment, and this project has been published in the Food Chem Toxicology journal. Then at the same time, I also model the DNA adduct formation based on a similar approach but different endpoint. Then aristolochic acids contain two types of congeners, either aristolochic acid 1 and aristolochic acid 2, and with this project we also want to measure how much the difference, because many cases saying that only AA1 that cause the kidney cancer or the urothelial cancer, but how about AA2? Then we also measure the difference between the DNA adduct formation of AA1 and also AA2. Then the final task for my PhD project, where I did the risk assessment of the aristolochic acids from the plant food supplements and herbal product that I sample from worldwide. So what I did was I measured the AE in the supplements, and then I did the estimated daily intake based on the requirement or suggestion from the supplier. Then I calculate the margin of exposure. Then based on this margin of exposure, then we can know that either this AA is need a risk management action or not. And this also project also has been published in 2017. During my PhD years, so I took an opportunity to present my work in many conferences and also in meetings. So this was the first meeting that I present my work in Portugal in 2015. Then I also presented the next year in the Dutch Society of Toxicology meeting. And this is right before my PhD defense, where I presented my work in the Society of Toxicology meeting in Baltimore, USA. I completed my PhD in March 2017, where I need to defend my thesis in public. This was me in the middle, where I need to defend my thesis. These were my opponents of during my thesis defense ceremony. So it was a great moment. It was really challenging. It was not easy, but I did it. So I think that was a very great moment for me, myself, and also for my family, where it's finally finished. So I think this is also a great moment for all PhD, where when they finish the PhD, or maybe they pass the Viva. Alongside with my PhD degree, I also took a lot of courses to be registered as European registered toxicologist. I got a lot of questions where regarding how did I get the certification. So I was very lucky to study in the Netherlands, where we have this course we call as a PET course, where this is education of toxicology, where I need to undergo a lot of courses. I think more than 10 courses that I attend throughout the 4 years of my PhD. Then after each courses there were exam, and then we need to pass the exam. And then after I completed my PhD, then I compile all the documentation that needed. For example, the current work, the papers that I have, and the contribution in toxicology. And then I apply this certification through Dutch Society of Toxicology. And then in 2018, I have certified as a European Registered Toxicologist of or ERT. So now from Europe to back home. So I was back in Malaysia in April 2017, and this was the first congress that I attend in 2017 for the during the third Malaysian Congress of Toxicology, where I shared my work. And then in 2018, I also present some work in the Asian Emerging Researchers Conference. And then in Bangkok, for the Asia Pacific Regional Meeting on Children Environmental Health, because my background is actually in environmental health together with toxicology. And then also attend a lot of training on occupational environmental health. And in 2019, I presented some work in IUTOX 11 International Congress of Toxicology, and we also was there to sign the agreement and also for promotion of this congress. Then supposedly in 2020, I was invited to attend the 59th annual meeting of SOT in California, but because of the COVID, the meeting was cancelled and changed to online version. And this is this year where I presented some work in the Young Scientist Network Asia Academy of Science Malaysia for virtual conference. And now I have different hats to wear, where my main role or my main responsibility is actually as a senior lecturer at Department of Environmental and Occupational Health, Faculty of Medicine and Health Sciences, University Putra, Malaysia. At the same time, I'm also an associate interim researcher at Natural Medicines and Product Research Laboratory, Institute of Bioscience, UPM. At the same time, I'm also a president of the Malaysian Society of Toxicology, a newly elected president. I was elected this year until 2023, so please pray for me. And at the same time, I'm also a treasurer of the Environmental and Occupational Health Society Malaysia, and I'm also an active member of Putra Toastmasters International. Now, let's move to the second part of my talk, where I will share with you the in vitro and in silico approaches in risk assessment. As we know that in risk assessment, we always need the animal data to derive the point of departure for risk assessment of chemical. But please bear in mind that what happened in animal, the prediction is not really it is not well predict the toxicity to human. So why we still need this animal study to derive the point of departure for risk assessment. So in this method, so what we need as an input is actually the in vitro toxicological effects, and what our goal is actually to predict what happened in vivo. Then how to translate this in vitro to in vivo is actually by using the computer modeling. And in our case, we use the PBK modeling based reverse dosimetry. So where what we need here is we need to understand the ADME of the chemical, the metabolism of the chemical, and then based on this metabolism, and then we put this data in the computer model, and we are able to translate from in vitro to in vivo to derive the point of departure of risk assessment. So in this presentation, I will summarize 6 strategies that you can use, you can do to plan for a risk assessment of chemical for any toxicological endpoints. Assessment. The first strategy is what you need to do is you need to establish the in vitro concentration response curve for the selected endpoint using the adequate in vitro model. So what I mean here is if you want to target for kidney, so you need to have in vitro kidney model, or if you want to focus on liver cancer or liver hepatotoxicity, then you should have a model for in vitro for hepatotoxicity. Or if you want to, for example, you want to model the brain tumor, so you need to have a proper model to represent the brain tumor. So what I did was, this is example of my project, so where this line, you can see from this red line is my data, and the gray line is actually from the literature. But from the literature data, we have a different cell that has been used to study the in vitro cytotoxicity response to aristolochic acid 1. So we can use this data to evaluate our model. Then the second strategy, when you have the dose response curve, the next one what you need to do is you need to develop the model to describe the in vivo kinetics of a parent compound and relevant metabolites in relevant tissue. So what does it mean by this is you need to understand how much and what is the metabolism of the chemical. For example, in my case, I have the AA1 or the AA1 as a parent compound, how much this chemical will undergo the activation and detoxification. So meaning that ADME, you need to understand the ADME of the chemical. So how much from this parent compound will activate and bind to DNA and form DNA adduct, and how much this chemical will be excreted from the body. Then after you understand about the metabolism, then you will quantify the kinetics parameter, for example, the formation of the metabolites in the specific enzyme. So for example, if this is in the liver and also in intestine enzyme in different model, and then based on this data, then you can put this data in the computer model. So for example, I want to model the oral exposure of AA1, and then once we expose through oral injection, and then it will go to intestine, and then it go to liver and go to venous blood, and it will circulate to the whole body. And then the metabolism happen in intestine and in the liver, so we will put some equations here, and then we eventually will evaluate how much the chemical will go into different compartment of the body. So the strategy number 3 is you need, based on the data that you have in the model and also the model, then you need to evaluate your model based against the literature data. So for example, this is the this is my model, and we also my PBK model of the aristolochic acid 1, and I compare the metabolism from different study, which is for example from Quo and also from Ren. And this is very important, the model evaluation, because you need to make sure that your data will is within the animal data that available in the literature. And then strategy number 4 is now you have the in vitro data and you also have the PBK model, so you can translate the in vitro concentration response curve to in vivo situation using the PBK model reverse dosimetry. So what does it mean by this? For example, this is what I predict of the in vivo dose response curve. For the acute kidney toxicity, I can use either Cmax or AUC, and I have also different cell lines. And then number 5 is based on the dose response curve that you have translated, then you can use this data to model the point of departure and you can use this data for the risk assessment. And number 6, you need to evaluate again your point of departure against the data from literature. So for example, these are data from different cells and also from different rat model, and then this is all from literature data, and this is our prediction. So what we conclude from this is our point of departure generally fall within the range of point of departure from in vivo study. So we know that we can use this approach to study the nephrotoxicity in vivo. As a junior scientist and also women in toxicology in Malaysia, so I face a lot of challenges, especially when I just came back from the Netherlands. Because as an academician, we have a lot of KPIs to achieve. For example, you need to be excellent, to excel in teaching, in research. You need to find a grant. You need to publish papers. You also need to do consultation, and also you need also to serve the community. So when you start, when I just came back, I feel like very overwhelmed with all the KPIs to achieve in 1 year. So, but I would like to summarize the challenges that I face in 3 different parts, which is the first one is about mentor. To find a good mentor is a big challenge for me. If you already have a good mentor, you are very lucky. You should be grateful for that, because if you have a good one, he or she will help you to put things into perspective and also to plan the future in efficient way. And if you still don't find one, please keep searching. The second one is about funding, because no money, no talk, and no money, no research. So meaning that here we really need money to run the experiment, especially in toxicology. We use a lot of money to buy chemicals, to buy media, and to buy a lot of things to run the experiment. So I was lucky, I have secured the FRGS, or the Fundamental Research Grant Scheme, in 2018, where I used this money to hire the postgraduate student to run the research. And also with the money, also I can use to buy chemicals, and also to go for a conference, and also to pay for the research article for publication. The next one is about collaborators, to have a good research culture in Malaysia, because I think to find a good collaborator is not easy, and how to find a good one is actually to try and error, because if you never work with someone, you never know how he or she works. So I think it's important thing to have a good research culture, where we have a good ethics, especially in research ethics. So I think these are the 3 challenges that I face when I just came back from the Netherlands. And if you are still a student, and if you would like to be in academia, so these are all the challenges that you might think and might face when you are in this line. So you already have, like, you can project what will happen and what you should plan for the future. But if you're already a senior lecturer, and I think please be a good mentor, and please help the junior to write a good proposal, so that they can have the money to run the project that they want. And also be a good collaborator, because I think if we work together, especially in Malaysia, because I think toxicology in Malaysia it's underrepresented. Many work, or many lab think that they do a good toxicological work, but if we don't collaborate, we never learn new thing. Now I will share with you my current work and way forward. So for now, I have few postgraduate students. The first one is Siti Soleha, where she worked with aflatoxin, the risk assessment of aflatoxin in the plant supplements in Malaysia, and she already like a final stage of her research work, and hopefully she will able to finish soon. The second one is Nur Azra Pauzi from National Pharmaceutical Regulatory Agency, where she worked with she continued my work. It's actually for using about a similar project but different endpoints. The third one is Muhammad Firdaus, where he also continued my work in aristolochic acid, but he will focus more on the exposure in Malaysia. So at the same time, I'm also applying for a grant to understand the molecular analysis of the toxicogenomics of the renal cancer patients and the implication of aristolochic acids in Malaysia. So we want to understand either are we exposed to these chemicals in Malaysia, and our cancer patients, are they exposed to these chemicals that could lead to that the cancer formation. So we would like to study this kind of effect. This is my research team for this project, where we have different background and very multidisciplinary team. So for example, we have a group of pathologists from UT Malaya, where they are focusing more on the sample collection from the cancer patient. So based on this human sample, we are going to extract the DNA and study the toxicogenomics of the sample. And with the help of geneticists and also bioinformaticians from International Agency of Research on Cancer in France, and also from Sunday University, and together with me from UPM. So we hope that we will be able to secure the grant, because we know that this study is important to understand the exposure of aristolochic acids in Malaysia, and also the implication, especially in the public health, to protect the public from exposure to aristolochic acids. Based on my presentation, you might think that I don't have life because I only focus on research and also scientific work. But I think you got me wrong, because I also enjoy my life quite a lot. So for example, this is the picture that was taken, I think, in around 2010 or 2011, when we had a dinner together with my friends and also the supervisors when I studied master in University Kebangsaan Malaysia. So this was me. I did a free fall or skydiving when I was in the Netherlands, and I also enjoy a lot of sports activities. I think this is a good way to channel the stress, especially for during my PhD years. And I also enjoy a lot of activities with a gathering among Malaysian Student Association in the Netherlands. So I came back, I also organized a lot of outdoor activities. For example, this was happened during before the COVID-19 pandemic, where we had a gathering with my mentees, and I think we need to have a good connection with the student, because they are all my mentee, and then if they have problem, they won't be they will just come back to me and then seek for help. And last but not least, I have a very strong support system from my family, because they are there since the day one of my life, and I know that we want to be a great scientist. We are striving to be a great scientist, a great toxicologist, a great teacher. But I think at the same time, we also need to think that we also need to be a great daughter, a great mother, or a great sister, and we also need to be kind to everyone. And when I say that we need to have a work-life balance, it sounds cliche, but I think everyone should strive to get a work and life balance. With that, I would like to thank all my collaborators, all my students for their hard work, especially during this COVID-19 pandemic. I pray that all of you will be able to finish on time. And I would also like to thank the Malaysian Society of Toxicology for giving me a chance to share my experience in toxicology in this Congress. And last but not least, if you are interested in toxicology, Don't forget to join us in the society, and I see you there. Terima kasih. Thank you.